So this is the note outline for chapter 8. I would pay particular attention to the vocabulary words because sometimes, you have to know the words, the language in order to be able to understand the concepts. And if you notice here, I've taken out the section, if you're following along in the textbook. The thermodynamics section is, I would say, concept 8.1. I think in the new book, it's 8.1 and 8.2. The part of the chapter about free energy and how to calculate free energy, that is not going to be part. The only thing that I've got here is this part: spontaneous processes or reactions, one that occurs without an input of energy in the system. It's not always fast. That is in the 10th edition of the book. That was all concept 8.2. I believe in the new edition, it might be 8.3. I'm not positive. That section is not on the note outline. So that means skip. Okay, don't study it. It's not going to be on the test. Not on the test, not on the exam. Don't study it, right? So if I express frustration when later you guys are like, do we have to know all that stuff about free energy? This will be why, because specifically it's not on the note outline. That means not covering it. And I'm not going to talk about it. That means not covering it. That means not on the test, okay? In Bio 1, it's going to be very rare that I'm going to just throw you to the wind and say, oh, you just have to study this whole thing on your own without any notes or any Zoom video for that, okay? It's Bio 1, right? So you need, I'm not trying to, say that you're not smart, but you need all the help you can get for this, because it's a lot of stuff in a short period of time, and it's all over the map as far as how it's relevant to biology, right? So I get it. That is a skip. Do not study the part about free energy and all of stability of reactions, not important. Delta G, skip, okay? The other parts on this note outline, you have to know all of them. So we're gonna start up here at the top with some general talk about metabolism and thermodynamics. And I'm gonna show you some pictures from your book as we go through and talk about them. You must know the laws of thermodynamics, the first law and the second law. And then we are going to spend a lot of time talking about the structure of ATP and about the bulk of the chapter is about enzymes. And this is the most important part because this is going to play into and lead us into the next chapter, both chapter 9 and chapter 10. Everything in those chapters is related to enzymes. And I'm pretty sure there is a lab exercise in your box also related to enzymes. So that'll probably be our first lab because it's relevant to what we're covering right now. All right, so I'm going to stop that share, and I think I'm going to try to get to, I think there's PowerPoints for chapter 8. And then I won't have to draw everything. So I'm just going to look here. No, doesn't look like it. Bummer drag. All right, looks like I'm drawing. Okay. All right then, so let's start our discussions about chapter 8. I usually start with something very simple and talk about the whole theme of the chapter is metabolism. And what that means is, unfortunately for you, it means all the chemical reactions in the cell are XN for reaction. And that's a lot of chemistry, right? Or a lot of biochemistry. We're gonna focus on 2 sets of metabolic reactions. One set are building reactions, which are more familiar to you because we've been talking in chapter 5. If you think about chapter 5 was all about building those macromolecules from the monomers. That's same. We're going to take those monomers like monosaccharides or an amino acid and build it into a polymer that then is going to have a function in the cell. Those building reactions have a special name. They're called anabolic reactions or anabolism. And that is when we take a small organic molecule or carbon dioxide in the case of photosynthesis, and water, and we convert it into a larger organic molecule. And the example that we're going to discuss for our anabolic reactions basically is all of chapter 10, which is photosynthesis. Okay. The catabolic reactions, this again, 2 words, opposite meanings, pick one, focus on it, and then if I ask you the other one, say, oh, that's the opposite of the one I know. Catabolic reactions are breaking down reactions. So that's when we take large organic molecules, like a polysaccharide, and we break it down into its monosaccharides. And we are going to focus on one. There are hundreds of these. We're going to focus on one catabolic reaction. And that catabolic reaction is the breaking down of glucose, or the oxidation of glucose, which is also referred to as cellular respiration. And that would be chapter 9, right? All of chapter 9. Now, these catabolic reactions usually release energy. And remember, the energy in cells is ATP, right? So they're going to produce ATP. Any reaction that produces ATP or releases ATP, we call that exergonic. And then you can, as you can imagine, right? Because these anabolic reactions were the opposite, they're going to require ATP to be put in. And because they require energy to put in, they're called endergonic. So that's where those 2 definitions of words come from. So I'm just going to write one of them here. I'm just going to put exergonic. And remember, then you can fill in the blanks on the rest of them. So that's part of the overview. Okay. The second part of the overview, so why we're studying this, right? As I mentioned, it has to do with energy, right? All total. And so that's why we have to talk about thermodynamics, because that is the study of energy conversions. And then the third part of the chapter is all about enzymes, because these are the proteins in the cell, in all cells, that perform, if you want to think of it that way, or that regulate the thermodynamics inside cells. So the cells are all about energy conversion. I need ATP to make a protein. Oops, now I have to break down some glucose because I need energy to build a different protein. So the cells are all about these energy conversions. And how the cell performs those energy conversions is by using enzymes. And enzymes are proteins in and of themselves. So this is the whole third section of the chapter that we need to talk about, and why we need to talk about enzymes, because they control all of the metabolism, okay? But by controlling all metabolism. So we're going to focus on photosynthesis as our building reaction because it's Bio1 and it's general. But anytime your body or your cells, any cell needs to build a membrane, needs to build a phospholipid, that's an anabolic reaction, that's a building reaction. Anytime a cell needs ATP for anything, that's a catabolic reaction, okay? So even thinking back to the membrane, all of those active processes, right? All that active transport, that requires ATP. That's going to come from the cell breaking down some kind of molecule, organic molecule, into a small organic molecule and releasing ATP. Like I said, we're just going to focus on the breaking down of one of those molecules of glucose. But obviously, you don't just eat glucose, and cells don't just import glucose, right? Go to chemistry. Go to Chem 2 if you're a nursing major, Chem 102 if you're a biology major. You're going to learn all about this in organic chemistry. You're going to learn all these names, these horrid names for all kinds of anabolic and catabolic reactions, okay? So it's coming, right, in your future. But for right now, we're just going to learn and start at the very basics, and we're going to start here with this energy of thermodynamics. We're going to start with that, okay? So let's see if I can just add a board. Can I do that? There it is. Okay, cool. Is the other one still there? Oh, yay. Okay. So let's go to the second board here and let's talk about the laws of thermodynamics then. Okay. So there are two. And the first law is super simple. The first law is energy can neither be created nor destroyed, only converted from one form to another. Okay. Short version, all energy for life on Earth comes from the sun. That's probably the one that you're more familiar with because that's the easy version, right? That's the version we teach you in 7th grade. Now, what does that mean? All energy for life on Earth. Okay, that means that energy, like I said, can neither be created nor destroyed, only converted from one form to another. Now, this is so important. That's why I'm bothered typing it all out. I know it's on your note outline. I know there's pictures of it in your book. This is really important. Now, what do we mean by one form to another? In the biggest sense, we mean converting potential energy to kinetic, or the flip, the opposite, right? Or kinetic energy into potential energy. That's in the biggest, broadest sense, that's what that means, right? We can't create nor destroy energy. That's what it means. All energy for life on Earth comes from the sun. Basically what we're saying here is plants do all the work. Plants, algae, cyanobacteria, other photosynthetic bacteria do all the work of converting light energy into chemical energy. Okay, so that's another way of saying this. Plants, algae, bacteria, photosynthetic bacteria, that takes too long to write, photosynthetic bacteria. They have the job of converting light energy into chemical energy. I just asked this question on my microbiology test. Energy in living systems is mostly composed of what type of energy? And the answer is chemical energy, because ATP is a chemical, right? So I know it's a lot of vocabulary, right? So let's start at this sort of general potential energy into kinetic energy. Most of you, I would imagine, have seen a picture, right, of a, in your book, they have a picture of a diver climbing up a ladder and saying that's when the person is converting, the climbing up converts the kinetic energy of muscle moving into potential or stored energy. And then when the diver jumps off the diving board into the water, that's converting the potential stored energy in the person into energy of action or motion. Those of you that have had more chemistry or more bio before, right, we usually say potential energy is stored energy and kinetic energy is the energy of action or the energy of doing work, right? And again, those are like the global terms, and all of the chemistry, all of the enzyme work that we're going to do later is technically, you can trace it back to either converting potential into kinetic or kinetic into potential. Now, this business here, where I said about the chemical energy, right? So I like to just call them types of energy. I know it's not super accurate, but I like to call them different types of energy to get you thinking about what kinds of energy the cell uses that chemical energy for, or that ATP for, right? The cell needs to do things like, let's say, chemical work, right? It needs to produce organic molecules and produce ATP. The cell also needs to do mechanical work or have mechanical energy to move things from one place to another inside the cell. So something that we didn't probably talk very much about in chapter 6, but molecules don't just float from the endoplasmic reticulum to the Golgi in order, those proteins, in order for them to finish being processed and sorted and labeled correctly and sent out to the cell. It requires ATP and literally mechanical energy along microtubules to move things, move vesicles from the endoplasmic reticulum to the Golgi, right? I would call that mechanical work. In humans, we think about big mechanical work, like, right, like moving muscles, that's mechanical work. Being able to sit upright, that's mechanical work. So chemical work, mechanical work, sometimes that mechanical work, we talk about it as transport work, right? I'm gonna leave it like this. Chemical, let's focus it back on electrical, because a lot of the chemical work that gets done in cells is actually electrical work, maintaining a membrane potential. And so we've got chemical, mechanical, electrical, something I'm missing, I think. It's not coming to me. Those are the big ones. Yeah. All right. Chemical energy, mechanical energy, those are the big ones. Electrical energy, those are the ones that we're going to see sort of over and over again inside cells. Now, what this means about converted from one form to another is just like it sounds. We can take electrical energy or light energy. So plants are going to be taking this light energy and converting it primarily to chemical energy that then cells are going to use to do mechanical or convert into mechanical energy. Now, that second law of thermodynamics, put that box over here. The second law says every time energy is converted from one form to another. Now, in global sense, that means from potential to kinetic or kinetic to potential. But super specifically, it might mean from light energy into chemical energy. So from light energy into ATP. Every time that happens in a cell, in the universe, right? Some energy is, we say, lost, or supposed to be parentheses, or released as heat. And heat is actually described as an increase in the randomness of molecules, molecule movement, let's say, right? And there's a name for that, it's called entropy. So in your notes and in your book, and sometimes in chemistry as well, you'll see this grand statement. Usually the second law of thermodynamics is stated as every energy conversion increases the entropy of the universe. Right? Sounds very blah, right? So I'm going to just write that. Sounds very grand and global. And this, what I've typed out here above, that's like the everyday, everyman sort of understanding of what that actually means. Now, why is she talking about this? I don't care about the laws of thermodynamics, right? No, of course, no one does this in biology. Except this second law, these two laws, every cell on Earth has to obey. These are laws of chemistry and physics. These are laws of the planet Earth, the universe, right? Our solar system. So every cell has to obey these laws in some way. And so the reason that we're talking about this at all is because cells, I want to say, deal with or manage these laws of thermodynamics by enzyme pathways, by metabolism, which are basically pathways. And this is super important because the pathway part, we have to have a pathway here, because if we just jumped from molecule A to molecule D, let's say, and I'm going to draw you a picture in a second here. If we just jumped from molecule A to molecule D, what would happen is we would create too much randomness in the universe, or too much heat, and the cell would die. And then we wouldn't be a cell anymore, and we couldn't do any energy conversions. Okay, so the idea of moving, breaking, or making chemical bonds in a pathway and converting one, back to the metabolism, right? Converting a large organic molecule into a small one, or a small one into a larger one. Cells are going to do that by enzyme pathways, and we're just going to take enzyme pathways. So the pathway means one chemistry, I'll say one chemical reaction at a time. And that's what enzymes do. They are going to be so super specific that each enzyme is only going to do one chemical thing. So I'm going to erase and draw on my board and try not to make it so that I turn off my volume again. I know that if I hit stop share, I think these go away, but that's all right. We'll just do it anyway. Put you back up here. Ready for my picture. It's a different time of day already, so you can get a little bit better focusing. I'm going to erase this transport business. And I just want to just foreshadow a little bit. When we talk about pathways, what that means is we start with, let's say, a square for molecule A, and the arrow is going to represent an enzyme or a step in the pathway. And these steps have to go in this order. And then to get to molecule B, let's say molecule B is, some of this has been cut off. And then there might be another step or another enzyme, we'll call it enzyme 2, to get us to, I don't know, let's make this smaller and with a circle. And then a third enzyme, which I'm just going to label enzyme 3, maybe to get us to a complete circle, right? So this is supposed to be kind of like that. It's like kind of like a D to an O, right? This molecule here looks very different than what we started with. But what I was trying to say to you about metabolism, pathways, and the laws of thermodynamics is I can't just jump from A to the last molecule here in one step, because if I did that, there'd be too much heat. Too much heat would come off from that single chemical reaction, and the cell would die. Okay, so cells are always going to do this chemistry conversion to maintain the laws of thermodynamics. They're always going to do them in these little baby steps, which is sad for you because you'll have to learn pathways that have lots of steps. Sorry, that's just the way it is. All right, so now I would like to talk to you a little bit about enzymes. Oh wait, looks like we're supposed to talk about ATP next. And the types of cellular work, type of work that cells need to do. And it looks like I'm going to have to draw ATP because I don't have my PowerPoints loaded ahead of time. Oh, shame on me. All right, oh look, they're still there, yay. All right, so I'm gonna try to draw, it might not be very good. I'm gonna try to draw an ATP molecule for you, because ATP is the main mechanism of how, ooh, I don't think this is going to go well, of how cells are going to, yeah, this is going to take me too long. Okay, would I expect you to be able to draw ATP? Nope. Would I expect you to be able to recognize it? Yes. That means if I put it as a picture on the test and said, what is this molecule? You should be able to tell me it's ATP. There's a double bond up here. Okay. Well, it's not horrid. I won't want to erase it though afterwards because I'll be so impressed with myself. Maybe. So what we're supposed to be noticing about this is that it is adenine, which is one of the nitrogenous bases that can be used to produce, let's be a straight line. That can be used to produce DNA and RNA. Right, so this is the A of adenine. So it's a purine, 2 rings containing nitrogen, and it's attached here at this nitrogen. We attach it to a sugar molecule, and I'll draw it in the same color that they have in your book. It's green. And the sugar in this case is going to be ribose. So it's a 5-carbon sugar, but notice the carbons aren't labeled. And I'm not going to put all of the hydrogens and oxygens on this guy because it's just going to take us too long. I'm too slow at this. And then the key portion here is that this 5th carbon, so 1, 2, 3, 4, this carbon, this 5th carbon sticks up. So this is where the CH2 is. That's the 5th carbon, and it's that carbon that the phosphates are attached to. I'm going to draw them in black. I put them out here, and actually I'm going to put them as yellow. They're always yellow. The phosphates are always yellow. Why? Because, well, it doesn't show very well. Let's make it orange. Because they're supposed to represent energy, and we think that it'll help you remember that they're energy because they are yellow like the sun, right? So that's what that's about. So I'm just going to put phosphate. And that's going to be attached to an oxygen and another phosphate and another oxygen there and another phosphate. There's the 3rd one. I'm out of room now. Okay, so this phosphate here, double bond O, right? Single bond O, so ATP, because there are, yes, 3, count them, 3 phosphates. Poorly drawn. Like some kindergartner has a hold of this pencil, but I do not have the digital motor skills that all of you have. Right, so, yay, that was a lot of work. Okay, so this molecule, we call it ATP because, and I don't know why, Colleen, but it sticks in my mind, right? Because you asked me that very first day, do we have to know all about ATP? And I'm like, no, not now, but now you do. So A for adenine, triphosphate, 1, 2, 3. This, breaking this bond here between phosphate 2 and phosphate 3, this is the bond that is going to release the most energy, right? So right here, this is going to release a lot of energy, high amount of energy. Oh, I should just go back to a text box there because that's way too much. All right. This one you can break, then it becomes ADP because it only has diphosphate. And then you can break off the next one, it becomes AMP as well. But this bond, breaking from ATP to ADP, is the one we're going to see most commonly happen in chapters 9 and 10. Okay, so that's the one we have to concern ourselves with. Now, why did I take all the time to do this? Because I need you to be able to keep recognizing these organic molecules. I need you to be able to look at this and say purine, and I need you to say a purine attached to a ribose and 3 phosphates, or at least 1 phosphate. Oh, that's a nucleotide. That's going to be used to make DNA and RNA. Oh, but if I add 2 more phosphates, now it's an energy molecule. So I want you to start to see that the cell keeps utilizing the same molecules over and over and repurposing them in different ways, okay? So that's why. Now, why do we care? What types of work? So ATP is the chemical energy that cells use to do work. So chemical energy in the form of potential, right, stored right now, it's a potential energy converted to kinetic when they're actually doing the work. And cells need to do three kinds of work. They need to do chemical work, they need to do mechanical work, and transport work. Those are the big three, okay? And I think there's blanks on your lab handout, on your lecture handout, because I wanted you to type those out or write those out yourselves. And then, of course, your book's going to give examples of the different types of work that cells are going to do. It's in a small figure box. They give you this giant chemical equation box. Don't worry about that. I'm not looking for specific examples of these, but I do think you should be able to tell me that these are the three kinds of work that cells need to use ATP for. And when cells use ATP for this type of work, they're converting the stored chemical energy into stored potential chemical energy into kinetic energy. And the work is always going to be mostly chemical bonding work, especially the chemical work is going to be making or breaking bonds. All right, now we can talk a little bit about enzymes. So where to begin, where to begin? That I think is better if we draw it on the board. So I'm going to go back over here. Because there's so many things I need to tell you about enzymes. Yeah, so I'm going to say for Wednesday, because I, like you,